germ disc. In the second week, what is going to happen, sir? Bilaminar germ disc will be formed. Now, as I told you, second week is actually very much difficult here. Like there's so many changes taking place. So I want you to be like more alert here. But at the same time, what I'm going to do, sir, I'm going to make it easy with the help of only 4 diagrams. Only with the 4 diagrams, entire second week will complete, sir. Okay? Come on. अब थोड़ा सा ना recall कीजिए, sir. Recall in your mind. Recall in your mind right now, guys. This is outer cell mass. Okay? And this one here will be the inner cell mass. And there is a cavity. And that is a condition of what here? Blastocyst. I mean to say the end of first week. First week वहाँ पर खत्म हो गया सर. अब आगे क्या होगा? This blastocyst will undergo implantation. तो जैसे implantation होता है, the moment implantation will occur, let us see the changes right now. So what will happen? Okay. The changes here are, first of all, the outer cell mass is the one which is going to become your trophoblast. Outer cell mass is going to form what, sir? Trophoblast. अच्छा, तो ट्रोफो outer जैसे implantation होगा, the moment implantation will occur, what is going to happen, sir? Outer cell mass is going to become trophoblast. Remember, that is also like the layer of cells only. It is also like layer of cells only, but we are not going to draw all the cells guys there. It will take a lot of time. So इसलिए just we are drawing it like a line there. But you imagine in your mind there, trophoblast. Is it okay? Trophoblast. Then what happens to the inner cell mass here right now, sir? Inner cell mass का क्या होगा? So imagine this blue colored one here to be your inner cell mass. See how much time it is taking to draw the cells and all, है ना? Now in inner cell mass, what is going to happen is only one layer of cells towards the cavity. So what I am trying to tell you is that not the entire, all the inner cell mass, sir. Only one layer of cells that is towards the cavity. Only one layer of cells which is towards the cavity here, that will be modified, and that will be modified into cube shaped cells. Only one layer of the cells is going to modify into cuboidal cells, cube shaped cells. And yes, this hypoblast, sir. And remember, my dear friends, hypoblast is the one which is actually going to become endoderm, sir. Hypoblast is actually the one which is actually going to become endoderm. Within few minutes, I'll prove you here that it is the one which is going to become your definitive endoderm. So because the hypoblast is going to become the endoderm, therefore, can I call this one as primitive endoderm? Yes or no? Yes, I can call this one as the primitive endoderm. Why? Because hypoblast is the one which is going to become your actual endoderm within few minutes. I'll show you, sir. क्या बात है, बढ़िया. Okay. I want all of you to remember it like a story, sir, one by one. Okay ना? तो यहाँ पर देखो, अब कहानी में वहीं से continue कर रहा हूँ. I'm continuing from the first week only. So there is a blastocyst. In the blastocyst, you have got outer cell mass, inner cell mass. Outer cell mass is going to become trophoblast, and now welcome to inner cell mass. One layer is going to become hypoblast. अब बढ़ते हैं आगे. Let's go ahead. Let us go ahead here. Okay, चलिए. So my dear friends, again tell me. I'm going to use the same colors here right now, sir. Okay. So what is this black colored line here? ये black colored line क्या होती है, sir? This black colored line here is nothing but your trophoblast, the outermost layer of cells, trophoblast. And this trophoblast is the one which is derived from outer cell mass. Inner cell mass is actually going to form cube-shaped cells. A layer with the cube-shaped cells. What is the name of this layer? Hypoblast. And hypoblast is the one which is also referred to as your primitive endoderm. Why? Because it is the one which is going to become definitive endoderm. क्या बात है. Now what is going to happen with the remaining blue colored cell? I mean to say remaining inner cell mass का क्या होगा? What will happen to the remaining inner cell mass? Remember, they are going to rearrange themselves into tall and columnar cells. They will rearrange themselves into tall and columnar. And what is this known as, guys? This layer is actually referred to as your epiblast. इसी को हम epiblast कहते हैं. Wow. And epiblast is the one, yeah, if it is hypo below, above it will be epiblast. And epiblast is the one which is actually going to become your ectoderm. It is going to become definitive ectoderm. So that is the reason why I can call this one as primitive ectoderm. Oh, this is primitive ectoderm. Kya baat hai, sir? Understanding? And please don't make a mistake. These two, sir, epiblast and hypoblast are derived from where only?
Inner cell mass.
These two are derived from where only? Inner cell mass. Keep that in mind. It is derived from inner cell mass. Badhiya, badhiya. Very nice. Very good, sir. So trophoblast will be derived from outer cell mass, and the inner cell mass is the one which is going to form your hypoblast and epiblast. हो गया. अब बढ़ते हैं आगे. कहानी को बढ़ाते हैं आगे से. Now, the epiblast cells are going to multiply. They are going to proliferate. These epiblast cells are going to multiply, and after multiplying they are going to form one layer of cells here, sir. Okay, and this layer of cells here is actually known as what, guys? Amniogenic cells. Amniogenic cells. Understanding here? So that epiblast is going to multiply to form a layer of cells known as amniogenic cells, and because of this amniogenic cells here, you are able to find a cavity here. What is the name of this cavity? This cavity is amniotic cavity. What is this cavity here, sir? Amniotic cavity. Kya baat hai? Beautiful, sir. And hypoblast will also not keep quiet. Even hypoblast will also multiply, and it will multiply to form a layer of cells here. Hypoblast will actually multiply to form a layer of cells, and this layer of cells here is going to be known as Husser's membrane. What is that one? Husser's membrane. Okay, and therefore because of that again a cavity is formed here, and this cavity is said to be the yolk sac, and it is the primary yolk sac. Obviously it is primary yolk sac. Lovely. It's like symmetrical diagram there. Okay, so epiblast will multiply to form amniogenic cells, cavity there, amniotic cavity. Hypoblast will multiply to form Husser's membrane, cavity there, and that cavity is yolk sac, primary yolk sac. Kya baat hai? Done. So from here, yes, my dear friends, remember in second week everything is like double. अगर आप देखोगे ना, in the second week everything is like double, sir. Okay, so it is like epiblast and hypoblast, two layers, amniotic cavity and yolk sac cavity. There are two cavities, and then again there are two layers, amniogenic cells and the Husser's membrane. Again there are two layers. So it's like, you know, double in the second week. Everything is like double. अब चलिए आगे बढ़ते हैं, sir. How many diagrams did I promise you in the second week? Entire second week we are going to complete in 4 diagrams, sir. So we are done with second diagram here. Now welcome to the third one. Now before going ahead with the third diagram, listen to me with all your ears open. Okay na? Kaan kholkar suno, sir. Okay na? Listen to me with all your ears open right now, guys. Arshad Ali, we cannot go to any previous slide now. Please, not only Arshad Ali, everyone I'm telling you, we cannot go to any previous slide right now. We are learning embryology. Let us just continue and finish that in a flow first of all. अगर हम आगे पीछे करेंगे, if you do like, you know, like again going back and coming forward, then it will be a lot of confusion here. Lot of confusion हो जाएगा. So let's go in a sequence. Let's go in a sequence right now, sir. Okay, now I'm telling you aram se slowly, and I'm even drawing the diagram with you, sir. Just be with me. That's what I'm telling you. Flow बिगड़ जाएगा. Condition right now. अब क्या होता है, ध्यान से सुनो, sir. Listen with all your ears open right now. Listen carefully now. Whatever this blue color is there here, this blue colored one is the one which is going to become your ectoderm. Whatever this red colored one is there, that is going to become your endoderm. If it is ecto and endo, common sense, in the middle what should be formed here, sir? Mesoderm should be formed here. क्या form होना चाहिए? Mesoderm. And that mesoderm is known as intra-embryonic mesoderm. क्या कहते हैं हम लोग इसको? Intra-embryonic mesoderm. It is inside the embryo. Actually, this blue colored and red colored one are the proper embryo. That is embryo proper. That is the only one which is actually going to become your baby. Understanding? So this is like intra-embryonic mesoderm. Apart from that, there'll be some mesoderm formed on the outer side also. And on the outer side of the embryo, this mesoderm is referred to as extra-embryonic mesoderm. अच्छा. तो दो तरह के mesoderm होते हैं. There are two types of mesoderm. Intra-embryonic and extra-embryonic mesoderm will be there. And try to make your life very simple here, sir. Don't confuse. I don't know why many students are confused. Keep it very simple. Extra-embryonic mesoderm will be formed during second week. I repeat again. Extra-embryonic mesoderm will be formed during second week. And intra-embryonic mesoderm will be formed during the third week. बस इतना ही याद करना है. This is the only thing that you have to remember here, sir. Okay. Tell me right now. Extra-embryonic का form होता है. When will be the extra-embryonic form? When will be the extra-embryonic mesoderm formed? Yes, that is in second week. And when will be the intra-embryonic mesoderm formed, sir? Intra-embryonic will be formed in the third week, sir. अब आप मुझे ये बताओ. Okay, now you people tell me, we are in which week right now? We are in which week right now? We are in second week. तो अभी कौन सा वाला form होगा? Now which one will be formed, sir? Intro or extra? Very good, Gopika. Very good, Sneha. Extra, है ना? तो चलिए आगे बढ़ते हैं. Now keeping this entire planning in mind, let's go ahead. And now what we are going to do is, I am not going to draw all these cells again and again. Let's make this diagram more simple. Come on. What is this layer, blue colored layer above? What is that blue colored layer above? What is this blue colored layer above? Epiblast. It will multiply to form what? Amniogenic cells. Cavity, amniotic cavity. Then what is this red colored layer of the cells below? Hypoblast. And that will multiply to form Users membrane. And what is this cavity? Yolk sac cavity. What is that cavity? Yolk sac cavities. तो आपको समझ में आ रहा है अभी? Are you understanding my diagram? So this big diagram here, those cells and all, we are trying to make it simple here, sir. Follow my diagrams here, sir. Yes. And then what is that black colored layer on the outer side? The black colored layer on the outer side is nothing but your trophoblast. है ना sir? Trophoblast. Let me draw it properly here. This is trophoblast. Chalo, whatever. Now, my dear doctors, why did I leave this space here? Why? Because यहाँ क्या होता है ना? Here there will be formation of extra-embryonic mesoderm. So whatever this yellow colored I'm highlighting here, you imagine that one as a mesoderm here. So कौन सा वाला mesoderm? Which mesoderm, sir? This is extra-embryonic mesoderm. Okay, done. Now, is there any MCQ possible here? Will they ask you anything here? No, sir. They will ask you here about the development of extra-embryonic mesoderm. From where are you getting this extra-embryonic mesoderm from? Remember, it is derived from yolk sac. It is derived from yolk sac, sir. Understanding? Extra-embryonic mesoderm will be derived from where? It is derived from the yolk sac. That will be asked in the exams. अब चलिए आगे बढ़ते हैं to the last and final diagram, sir, the fourth diagram. हो गया बच्चों?